Greetings to all. Let us talk one more topic under biochemistry, that is lipoproteins. So what are the objectives of our presentation? So we need to define what is lipoprotein, explain structure of lipoprotein, and we have to discuss regarding types of lipoprotein and functions of lipoprotein. So lipoprotein is a biochemical compound that consists of both protein and lipids. So it's a biochemical compound which consists of protein and lipids. And usually fat absorbed from the diet and fat synthesized by the liver. So what is happening here? Fat is absorbed from the diet and fat synthesized by the liver. And adipose tissue must be transported between the various tissues and organs for utilization and storage. So what is the meaning of synthesis? The combination of components to become whole. The small molecules all togetherly will forming as the whole structure, that is known as the process of synthesis. Since the lipids, so what are the lipids? Triacylglycerol, cholesterol and cholesterol esters and phospholipids are insoluble in water. So these are insoluble in the water. These lipids are insoluble in the water. And the problem arises, transport them in an aqueous environment of the blood plasma. So these are insoluble in the blood. So in this manner, what is happening? It is unable to transport. It needs a carrier. So that is what they are discussing. They are explaining here. Here the problem arises to transport them in an aqueous environment of blood plasma. The lipoproteins solve the problem of transporting fat in the plasma. So these are lipoproteins helping as a carrier. These lipoproteins solve this problem of transporting fats in the plasma. Lipids are transported in bloodstream as lipoproteins. So these lipids are transported in the bloodstream as lipoproteins. Lipoproteins are large water-soluble molecules. These are large water-soluble molecules or complexes which are formed by combination of lipid and protein. Lipoprotein transport insoluble lipids through the blood between different organs and tissues. So what is this lipoproteins are doing? These lipoproteins transport insoluble lipids through the blood between different organs and tissues. So let us identify here the structure of the lipoprotein. So lipoprotein consists of a lipid core containing non-polar triacylglycerol which not having any positive or negative energies. These are that's why we are considering as a lipoprotein is consist of a lipid core. It's a lipid core containing non-polar triacylglycerol and cholesterol ester surrounded by a single layer of amphipathic phospholipid. This is a single layer. So we can see this monolayer or amphipathic lipid, phospholipid and free cholesterol. Triacylglycerol and cholesterol ester surrounded by a single layer of amphipathic phospholipids and free cholesterol molecules with some proteins. This is known as apoprotein. So some of the proteins, these are some of the proteins. This is a peripheral apoprotein. Then here we are notifying as a monolayer of amphipathic and lipid, phospholipid and cholesterol, free cholesterol. And this considering as a phospholipids, and these are known as cholesterols, free cholesterol and core of non-polar lipids. This is a core of non-polar, that is like cholesterol ester and triacylglycerol. I can see the picture. We can see this is a structure of lipoprotein. And what do you mean by CE? CE means cholesterol ester and TG is indicating triacylglycerol. And the protein components are referred to as an apoprotein or apolipoprotein. These protein components are known as apoprotein or apolipoprotein. And there are four major types of apolipoproteins. These are designated by letters A, B, C and E. So let us talk about regarding types of lipoprotein. Lipoproteins have been categorized into four major classes. How many classes? Four major classes according to their physical and chemical properties. The lipoproteins can be spreaded by ultracentrifugation. Separated by ultracentrifugation. It's a process. So one of the devices identifying in the laboratory that is known as centrifuger. With the centrifuger, we can able to separate this lipoproteins. The lipoproteins can be separated by ultracentrifugation. So ultracentrifugation, what is the meaning of this ultracentrifugation? A high speed centrifuge used to separate colloidal solutions. So what do you mean by colloidal solution? Colloidal solution means blood related products. The main types of lipoproteins are the first one, mainly we having four types: chylomicrons, very low density lipoprotein, which is denoted by VLDL, low density lipoproteins LDL, high density lipoprotein HDL. So let us talk about regarding chylomicrons. So chylomicron, a small fat globule. Chylomicron is a small fat globule which is composed of protein and lipids. And the lipoprotein complexes contain different portions of lipids and proteins. The density of these lipoproteins is inversely proportional to triacylglycerol content. So how it is managing? The density of these lipoproteins is inversely proportional to triacylglycerol content. As the concentration of triacylglycerol decreases, density will be increases. Oppositely, the concentration of triacylglycerol is decreases and density will be increases. What do you mean by density? It is a mass of a unit or volume. As the density increases, the diameter of the particle decreases. So chylomicron containing about 2% of protein and 98% of triacylglycerol have the low density. So whereas HDL containing 55% of protein and 45% of lipids has the highest density. Then triacylglycerol is a predominant in lipid and chylomicron and VLDL. So cholesterol is a predominant lipid in LDL, whereas phospholipid is a predominant lipid in the HDL. Now you can see the picture of this diagrammatic representation of lipoprotein with increasing densities. So here chylomicron is a large one and VLDL chylomicron is a very large, VLDL is a large one and small one is LDL, smallest one is HDL. So here density increases, percentage of protein increases and diameter is decreases. So let us notify regarding functions of the lipoprotein. Each class of lipoprotein has a specific function depending on their site of synthesis, lipid composition and apolipoprotein content. The first one is the chylomicron. Chylomicrons are synthesized from dietary fats in the small intestine. Where it is going to be synthesized? It is synthesized from the small intestine. They carry dietary triacylglycerol, cholesterol and other lipids from the intestines to the peripheral tissue. Intestines to the peripheral tissue. Then second one is very low density lipoprotein, that is known as VLDL. Very low density lipoprotein is synthesized in the liver. It transports triacylglycerol from liver to peripheral tissues. And third one is low density lipoprotein, that is also notifying as a LDL. Low density lipoprotein is synthesized from plasma VLDL. Plasma VLDL. VLDL means very low density lipoprotein. It is a major carrier of cholesterol in blood. So this is a major carrier for cholesterol in the blood. The role of LDL is to transport cholesterol from liver to peripheral tissue. So what is happening here? It is transporting cholesterol from liver to the peripheral tissue and regulate the cholesterol biosynthesis. That's the reason we notifying as a LDL cholesterol is known as bad cholesterol. Then the fourth one is the high density lipoprotein, that is also known as HDL. This high density lipoprotein synthesized in the liver and small intestine. It transports cholesterol from peripheral tissue to the liver where it can be catabolized and excreted. It is the both or vice versa. Whereas HDL HDL is transporting cholesterol from peripheral tissue to the liver, where it can be catabolized and excreted. Whereas in the LDL, LDL will be transporting cholesterol from liver to the peripheral tissue, and the HDL will be opposite, peripheral tissue to the liver. Then HDL cholesterol is called good cholesterol. The process of transport of cholesterol from tissues to the liver is also known as reverse cholesterol transport. What is the meaning of this? Reverse cholesterol transport. Why we are considering as it is a reverse cholesterol transport? The process of transport of cholesterol from tissues to the liver. So opposite it is going on, tissues to the liver. That's the reason we notifying as it is also known as reverse cholesterol transport. Then what's the difference between good cholesterol and bad cholesterol? HDL cholesterol is considered to be the good cholesterol because it removes excess cholesterol. It removes excess cholesterol from peripheral tissue and transport it to the liver where it is degraded or excreted in the liver. So thus HDL tends to lower our blood glucose level. Blood cholesterol level. So why we are considering as this cholesterol, HDL is a good cholesterol because it helps to lower our blood cholesterol level. As I told you in the previous classes, the normal blood cholesterol should be below 200 milligram per deciliter. On the other hand, LDL cholesterol is called bad cholesterol because it transports cholesterol from liver to the peripheral tissue. It transports the cholesterol from liver to the peripheral tissue. For excretion, cholesterol must enter the liver. Always remember that the cholesterol must be transported to the liver for excretion. But LDL, what it is doing? LDL is doing like it is sending cholesterol from liver to the peripheral tissue, whereas HDL is helpful for the cholesterol shifting from the peripheral tissues to the liver. The risk of coronary heart disease, that is CHD, is related to plasma levels of total cholesterol and LDL cholesterol. And let us summarize our topic. So far we have discussed regarding lipoproteins and its structure and its function. So what do you mean by lipoprotein? It's a biochemical compound that consists of both protein and lipids. So structure of lipoprotein, basically it consists of the non-polar structures and types of lipoproteins. We having that chylomicron, VLDL, very low density lipoprotein, LDL, low density lipoprotein, and HDL, high density lipoprotein, and these are having each one having their own functions of lipoprotein. Mainly it will for the carrier because these fats are insoluble in water, so it requires some carrier. So that carrier is the lipoprotein is helpful as a carrier for transporting of fats in the aqueous environment. I hope my dear students you understood the concept of the lipoproteins. Have a nice day. Once again we will meet.